Hello students, today we are going to start the next part of the nervous system. The introduction of the nervous system was already completed in the structural organization in animals. The tissue, whenever we have studied, we have already completed the introduction of the nervous system. We have seen the neuron, the types of neuron that are apolar, unipolar, pseudo-unipolar, bipolar and multipolar neuron. Then a TS of nerve and then the structure of the neuron we have already completed. After that we will see the cells of the nervous system. There are different type of cells. Actually, basically we can divide the nervous system into the cells of the nervous system into two types only. One is the neuron and second one is the supporting cell. So what are the supporting cells? They are non-excitatory cells which are helping the nervous system in many different ways. Already I have shown here. So you can see the excitatory cells are the neurons, okay, who can transmit these are the excitatory cells which can transmit the nerve impulse from one neuron to the next neuron. But if it comes to the neuroglial cells or the supporting cells, they are the non-excitatory cells. Okay, non-excitatory cells means they cannot transmit the impulse. Okay, they cannot transmit the impulse from one neuron to the next neuron. They are supporting the nervous system. Actually, more than 50% of the cells of the nervous system are the neuroglial cells itself. Okay, so remember this one also, that is more than 50% of the cells are the neuroglial cells. Now we will see that a different type of neuroglial cells we have. What are they? We have seen here these are ependymal cells, then microglial cells, then astrocytes, then we have the Swan cells, and another we have that is oligodendrite, but the function wise they are same. So first of all, just see what is the ependymal cells. What I have showed here, that they produce the CSF. It is not the only production, but it will also absorb the excess of CSF, as well as it will move the CSF too. Okay, so CSF means you already know, that is cerebrospinal fluid. Cerebrospinal fluid is one specialized type of fluid which is formed from the blood itself, but this CSF will not have any type of impurities, toxic or drug materials in it. So it is the purest form of blood which will transport the nutrients and oxygen, the gases, to the brain. Okay, so here the CSF production, the CSF, that is cerebrospinal fluid, is formed freshly in every after 24 hours. So every day it will produce CSF. Excess of CSF will be absorbed also by the ependymal cells. And the third thing is it will also keep the CSF in a moving state. Okay, so how the CSF look like? The CSF, it is a type of columnar cell. Okay, so remember this is one specialized type of cell. The columnar cells have the nucleus towards the base. This is already you know. They have the brush bordered columnar cell, and another thing you also need to know, that towards the beneath base it is also going to have the ciliated lining. Okay, so it is going to have brush bordered above apical region and ciliated base region. So what is the function of this brush bordered epithelium, columnar epithelial cell? That it is going to absorb the excess of CSF. If it is ciliated beneath, that reason is that it will keep this CSF in a moving state, and also it will secrete the CSF by itself. Okay, so this is the ependymal cell. Next thing, that is the microglial cell. This is important. It is the phagocytic cell. So suppose this is the microglial cell. Okay, so this microglial cell will take, or it will absorb, or it will destroy any type of foreign particle. If it is suppose a foreign particle present, it will take the foreign particle first engulf it, then after that it will absorb it, and after that it will destroy the foreign particle too. So what is this? This is microglial cell. The main function of this microglial cell is phagocytic. So it is like the monocyte and monocyte as well as the macrophages. It is like the function of monocyte and the macrophages. It is having in one function that it will kill the foreign particle if it enters or it is present in our brain, spinal cord, and as a whole in the nervous system. Next one, the third type, that is the astrocytes. So if you see this astrocyte, how they look like? Okay, so one thing you can know, astro means it is star shaped, cyte means it's a normal cell. So it is star shaped like this. Okay, so it is star shaped cell. So what is it? This star shaped cell, astrocyte, it will form or it will heal or it will repair any type of tissues, nervous tissues, if it is dead or degenerated, that will be repaired or healed by the astrocytes. Another very important function of this astrocyte is to form a blood-brain barrier. Now what is the blood-brain barrier? Suppose this is your brain, okay, and this is your blood vessel. So here this is the blood vessel. In between the brain and the blood vessel, we will have a lining of the astrocyte, and this astrocyte will be selectively allow the things that will enter inside the brain or not. See, what happens in our body, the nervous tissue is the only tissue who cannot divide. The mitotic division will be not possible in case of neurons. So if some neurons destroy or die due to any infection, that will be not possible for us to happen. So that's why to make the neurons safe and secure, their infections has to be decreased. So that's why through the blood vessel, if the blood vessel have any toxic material or any dangerous or harmful elements in it, that will be not allowed to enter into the brain. So this will be functioned by the astrocytes. That's why it is called as blood-brain barrier, that will selectively allow only some particles, that is nutrients and gases, that are important molecules, to enter inside the brain, but the harmful elements will be not possible to enter in the brain. Okay, so that's why infections in the brain is very rare. So very little case we can see that infection of the brain has happened. Okay, it's very rare in this case because blood-brain barrier, astrocytes are there. So that's why if anyhow any infections are there, antibiotics are not going to work easily on the brain. So for that, very powerful antibiotic will be needed, because this antibiotic will be also not able to allow to enter in the brain easily because of the presence of astrocytes, right? So this is the astrocyte. Okay, so the next one, that is the Swan cells or oligodendrites. These are actually two different type of cells. The Swan cells or oligodendrites, they will produce the myelin sheet, but actually it's not like always they will form the myelin sheet. So if the Swan, suppose this is the axon, around the axon, just suppose the Swan cell, and this Swan cell is innervated by the axon, then it cannot produce the myelin sheet. Okay, if around this axon, if there is a coiling or wrapping of the Swan cell occurs, then only it is possible it can form the myelin sheet. Remember, when the myelin sheet can be produced, if axon innervates, okay, if the axon innervates the Swan cell, then it cannot produce the myelin sheet. If around the axon, the Swan cells make or wrap around the axon, then only it is possible that it can form the myelin sheet. Okay, so this is one very important point you need to know. Next thing you need to know that if you see Swan cells and oligodendrites, this is going to be very easy. You need to know that it is going to form the myelin sheet. But the Swan cells will produce the myelin sheet in the peripheral nervous system. It will form the nerve that myelin sheet in the PNS, peripheral nervous system, and if it is oligodendrite, it will form the myelin sheet in the central. So this is in the brain and the spinal cord. Okay, so Swan cells will form the myelin sheet in the PNS, peripheral nervous system, and oligodendrite. The same similar type of cells will form the myelin sheet in the central nervous system. So if you can recall the nervous system, what is the origin of the nervous system? It is ectodermal in origin, right? So it is, let me switch this pen. So it is ectodermal in origin. Okay, so this is ectodermal in origin. Okay, but one thing you need to know, there is one exception in the microglial cell. What is that exception? This monocytes or lymphocyte type of cells are microglial cells. So it is derived, these are the cells of the connective tissue. So what is this? This is mesodermal in origin. Okay, so remember this thing. This is only the exception that dermal. Okay, so what is the exception? Exception is in the origin. So all of the cells, the neurons, neuroglial cells, all of them, they are ectodermal in origin except the macroglial cells. The macroglial cells, they are actually mesodermal in origin. So after that, let's just move to the next part. Okay, so after that, let's just move to the next part, that is the types of neuron. Already we have seen one part, that is the neurons. There are 5 different types depending on structure, that are apolar, unipolar, pseudo-unipolar, bipolar and multipolar neuron. So that part already we have completed. So after that, let's just move to the next part, that depending upon the myelin sheet, also the neurons are of 2 types. Okay, the neurons can be myelinated neuron. Okay, it can be either myelinated neuron or it can be non-myelinated neuron, right? So if myelin sheet is present, then it will be called as the myelinated neuron, and if it is non-myelinated neuron, then it will not have the myelin sheet. But does not mean that I'm saying that Swan cells are present or not. Swan cells can be present in the non-myelinated as well as myelinated cells. Okay, so what is happening in the myelinated nerve, or that neuron which have myelin sheet, that has the Swan cells which wrap around the axons and form the myelin. But if it is non-myelinated type of neuron, they may have the Swan cells, and these Swan cells are getting innervated by the axons, and that's why the Swan cells cannot form the neuron. The Swan cells cannot form the myelin sheet. So that's why it is called as non-myelinated type of nerve, or that nerve formed by the neuron, that will be non-myelinated neuron. Normally, myelinated neuron will be present in the CNS, that is making the impulse, the movement of impulse faster. But in case of non-myelinated neuron, that is present in the PNS, which makes the nerve impulse to go a little slower. Okay, so that is the myelinated and non-myelinated type of neuron. Then after that, function wise also the neurons can be of different types. Okay, so let's just see. If it comes to the function wise, we have 3 different type of neurons. What are they? Sometimes it can be sensory neuron, sometimes it can be called as the interneuron, and sometimes it will be called as the effector neuron. Okay, so just see what happens whenever a stimulus is generated. Okay, whenever any type of stimulus is generated, it can be, suppose you smell something, you hear something, you are touched. So any type of stimulus, if it is generated, then what will happen? A receptor organ are going to receive it. Receptor organ means it is one kind of a sensory organ. So if it is smell, then nose. If it is a vision, a light, then eye. If it is a sense of any touch, pressure or temperature, that will be skin. So the receptor organ will be the organ which is taking this stimulus. So this receptor organ will take this sense, and one type of neuron will help in taking that stimulus via this receptor organ to the brain. Okay, to the brain. So brain or spinal cord, we can call it as a CNS. Okay, so CNS, this is the CNS which is taking that impulse. So via one type of nerve it will go, that will be the sensory nerve. And sensory nerve will be also called as the efferent nerve. Okay, this is called as the efferent nerve. So which is made up of efferent nerve, that will be the sensory neurons. Okay, so the sensory neurons will take this sense from the receptor organ to the central nervous system. Now the central nervous system is going to have some type of neuron also, that will be called as some interneurons, or you can call it as the relay neurons, or it can be called as the associate neuron. Okay, so these are the neurons which is present in the central nervous system itself, who can able to receive the sense or the stimulus from the sensory neuron via a nerve. That nerve is called as the afferent nerve. Then after that, CNS will make a response of it, and whenever the response are produced, that response will be taken by another type of nerve. What is that? This is called as efferent nerve, and this efferent nerve will take the response. Okay, so we are producing a response. So response will be produced by the central nervous system via the efferent nerve, which is made up of a neuron that is called as the effector neuron. So effector neuron will take this response via the efferent nerve, and where it will go to the effector organ. This effector organ which is going to produce the response. Okay, so this is about the types of neurons we have. Okay, already one type of neuron we have already seen depending on structure. Now we are going to see that the types of neuron, depending upon the presence of or absence of myelin sheath, you have myelinated neuron, or you may have the non-myelinated neuron also. Second thing, that if it is function wise, we have three types of neuron: sensory neuron or afferent neuron, you can say. Now in the central nervous system, we can have relay neuron or interneurons. Then last one, we have the neuron which will produce or take the response. Take the response. That will be called as the effector neuron. So that is all about the parts we have seen in this video. In the next video, we will see that whenever the stimulus is taken up by the receptor organ, then how this afferent nerve will take this stimulus and it will go to the central nervous system, or how the central nervous system will transmit this impulse to our effector organ. So in the next video, we will see the conduction of nerve impulse within one neuron. So this is up to this video we have completed. So I hope you understand. Thank you.